GSE159751 is an RNA-Seq FPKM dataset comparing Crohn's disease patients with non-IBD controls. Although FPKM is normalized per 1 million reads and per 1 kilobit, the distributions remain varied. So we recommend applying global normalization after removing data lack. But global normalization is a linear method, which means the distribution shapes remain unchanged. You see the shapes fall into two types: monomodal and bimodal. The monomodal shape often indicates RNA degradation. Hierarchical clustering shows that the samples are separated into CD and control clusters. But if you look more closely, each cluster is divided into two subclusters, representing mono and bimodal distributions. Of course, the mode type depends on an experimental factor, not a biological one. You can say the CD and control samples are stratified by the outer factor. You can select the monomodal subclusters from the CD and control groups and view them on the PCA chart. The first principal component captures the difference between the control, right, and the CD, left. On the other hand, the likely technical bias forms the second principal component. The difference between CD and control is clearer when comparing only the high-quality, bimodal, samples. You see that monomodal subclusters introduce noise, disrupting differential expression analysis. We have lists of differentially expressed genes between the CD and control groups using all samples and only bimodal samples. If you compare the up-regulated genes in the CD group, you can get more candidates by using only bimodal samples. The number of down-regulated genes are almost the same. Anyway, you see that removing noise samples can bring more opportunities for discovery. By the way, bioinformaticians have introduced several sophisticated methods to remove nonlinear systematic biases, such as differences in distribution shapes seen in this case study. Quantile normalization is one such technique that makes the distribution shapes uniform. You may think, that's great news. However, wise experimental biologists must be careful to test whether it works on their own. Currently, having a good experimental design and delivery plan to generate high-quality raw data and tools to monitor what happens is superior to relying on sophisticated algorithms.